Hello, my name is Lincoln Nadald. I'm a medical oncologist and physician, and I'm medical director of the Cancer Genomics program at Intermountain Healthcare. And today I wanted to spend some time talking about what is cancer genomics. In this world of personalized cancer medicine and targeted treatments, the question that frequently arises is, what is cancer genomics and what does that mean? Let's just talk about that for a minute. This is my rendering of a human cell, and there are trillions of these throughout our body. We have liver cells and lung cells and skin cells and all different cell types. And what they all have in common is at the center are these long strings of DNA, and these are called chromosomes. In total, there are 46 chromosomes in every cell of the body. If we were to take one of these chromosomes out and stretch it out tightly, we would find that all along there are these discretely organized regions called genes. So genes are just little pieces of DNA organized along a chromosome. If we were to add up all of the genes across each one of these 46 chromosomes, we would find that there are approximately 20,000 genes. And it's the same 46 chromosomes and 20,000 genes that are present in every cell of the body, every lung cell, every liver cell, every skin cell. This complement of DNA with its 20,000 genes and 46 chromosomes is termed one genome. There is a copy of the genome in every cell of the body. Now, what do genes do? Well, these genes all have different functions. An example would be one called insulin. Many are familiar with insulin. Of course, it's a big deal for diabetics, for example. Genes also can do very mundane things like tell our eyes what color to be. So there might be a gene that says our eyes should be blue, for example. Then there are other genes that have very important functions, like telling our cells when it's time to grow and divide. An example that I like to use is a gene called KRAS. It's kind of a funny name. At the right time, a gene such as KRAS would get turned on in a cell, and it would tell that cell that it's time to grow and divide. And that's what would happen, and then we have two daughter cells that have been formed, and then the gene would turn off. By doing that, we've accomplished something very important. We've replaced any cells that might be dying and sloughing off. So this happens on our skin all the time, for example. We have cells that die and slough off, and to replace those, our cells will turn on a gene such as KRAS. These cells will divide, and then the gene turns off. The problem in cancer is that a gene like this can acquire an error or a mutation at some point during our life. And if that happens in a gene such as KRAS, then when it turns on, it will tell a cell to grow and divide. But now because of this error or mutation, the gene cannot turn off, and it will constantly tell these cells to divide, 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 and they will constantly divide and never stop because of this altered gene. And pretty soon a mass forms and we have a tumor, and that's where cancer comes from. At its very core, cancer is a disease of faulty or mutant genes. Well, what about cancer genomics? The study of genomics means the study of someone's chromosomes and genes. Cancer genomics means we now have the ability to take the DNA and genes out of someone's cancer cell and look for the list of faulty genes or mutant genes that cause that cancer to grow. And when we do that, we usually find that there are 2 or 3 or 4 faulty genes in each tumor. And they might have unusual names. I mentioned the one called KRAS, and there are dozens that I could list here. Some examples might be HER2 or FGFR1 through 3, or mTOR, on and on the list goes. The names don't matter, but what matters is that we now have drugs that can just target one gene or another if we can just know what the list of genes is. And so cancer genomics involves taking DNA from someone's tumor, identifying the list of gene alterations, and then using that information to provide targeted therapy, which can open up new avenues of treatments for patients who have run out of options. And so that, I hope, clears up some of the questions around what is cancer genomics.